Good evening, ladies and gentlemen. Welcome to Newcastle University. Welcome to the latest in the Insight Public Lectures. My name's Andrew Blamire. I'm Dean of Translational and Clinical Research here in the Faculty of Medical Sciences, and it's a pleasure to welcome you to tonight's lecture. Before we get on and I introduce the speaker, there's a couple of housekeeping things. We're not expecting any alarms to go off, so if the fire alarm does go off, we would have to exit the building in the way that you've come in. Very unlikely to happen. I would, though, ask that if you've got a mobile device that you would switch it to silent in order that we don't disturb the presentation and the flow of the evening. There'll be plenty of time for questions when we get to the end, so when those things occur to you as you hear the presentation, store them up and we'll have a good discussion hopefully at the end of the session. So, it does give me great pleasure to introduce a close colleague of mine, Professor Volker Straub. He is, I have to get the official notes to make sure I get all of the many things that I need to tell you about Volker. So he's the Harold Macmillan Professor of Medicine, Professor of Neuromuscular Genetics. He's also the Deputy Dean of the Translational and Clinical Research Institute here in Newcastle. That's his kind of local role. The other big local role is that he founded and is the director of the John Walton Muscular Dystrophy Centre, which is indicated on the slide, which is based at the Centre for Life, one of the biggest and I think most successful research groups in the medical faculty at the current time. He's also internationally renowned and internationally sought after, so it's great that we've got him here speaking tonight. He's chair of the World Muscle Society. He's involved in endless numbers of international trials all around neuromuscular research. And that really reflects what I know from having worked with him, that he's driven by a passion to understand neuromuscular diseases and to take that understanding and turn that into clinical trials that will improve patient benefit, patient quality of life, and patient outcome. He's absolutely the epitome of a translational researcher. I think I should probably stop waffling on and take the opportunity to welcome Volker and hand over to him. He's going to be telling us about the Newcastle way, leading advancements in neuromuscular research.
Thank you. Thank you very much, Andy. That was really lovely, I have to say. I would also like to welcome you. It's great to see public interest in the work we do here at Newcastle University, and in our case also across the Newcastle Upon Hospitals NHS Foundation Trust. I start with a disclosure statement because I will mention a few drugs in my presentation. I work very closely with industry, pharmaceutical companies, biotech companies. I don't hold any shares and none of them have influenced my talk in any way. So it's still important. Now, Andy has already introduced myself, and I'll also do a kind of introduction because, you know, what is my background actually? I was trained as a physician, and then I specialized and became a pediatrician. And then within pediatrics, I specialized to become a pediatric neurologist, so dealing with neurological diseases of childhood. And then within pediatric neurology, I was interested in neuromuscular diseases. I'll tell you more about what that means. And within the neuromuscular diseases, I'm really interested in muscle diseases, and I'm sometimes referred to as a myologist because myo is muscle. And among the muscle diseases, I'm interested in muscular dystrophies. These are muscle wasting diseases. And there are about 50 different forms of muscular dystrophy. And I'm particularly interested in limb girdle muscular dystrophies. I'll tell you a little bit more about this. And among the 34 different forms of limb girdle muscular dystrophies, I'm interested in the dystrophin glycoprotein complex associated limb girdle muscular dystrophies. So I'm a real geek, a super specialist. And I came to Newcastle about 21 years ago, and this was my application photo at the time. And you've seen times haven't always been easy. And Andy mentioned it. I'm also, this is actually Harold Macmillan in his chair. And I became the Harold Macmillan Chair of Medicine. And because Harold Macmillan was actually Earl of Stockton, the catchment area that I'm working in is across the Newcastle Trust and the North Tees and Hartlepool Trust. Now, I've mentioned that I'm a super specialist, and you know, this is a public lecture, and you are, from my perspective, generalists when it comes to my expertise. So I hope that we find a way to communicate and that you understand me. Now, my big challenge is also to make sure that no one is falling asleep, of course. And so the title of the lecture is The Newcastle Way, Leading Advancements in Neuromuscular Research. And you could say, well, why Newcastle and why neuromuscular research as a public lecture? There are now many databases that you could ask about rankings of scholars, academics, and of institutions. And I've picked Scholar GPS because it holds information, scoring, and ranking about 30 million scholars in 50,000 academic institutions. And then you can search for neuromuscular diseases, institutional ranking worldwide. Okay? And what's great to see is that Newcastle University is seen as the top university in the world when it comes to neuromuscular research. Of course, many people in the Northeast don't know about this because most people in the Northeast never heard about neuromuscular. Only if you are affected by one. But we have patients actually from all over the country coming to us. And there's a certain gratification to see that London and Oxford are ranked below us. Now, this ranking also said lifetime neuromuscular. And when it's lifetime, I have to admit that's not me because we're top. I came here because Newcastle is top. And this was actually founded by two eminent colleagues, Frederick Nattrass and John Walton. And Frederick Nattrass was the mentor of John Walton. And Frederick Nattrass was a neurologist. He was working for the RVI. He was actually the first whole-time chair of medicine in Newcastle. And he did a doctoral thesis for his MD on neuromuscular diseases. And a young colleague was coming to him and said, could you give me some advice what I should research on? And he said, why don't you look at myopathies, muscle diseases? And Frederick Nattrass was brilliant, but he probably didn't know how amazing John Walton was because he asked him to do this, and John did this very thoroughly. He wrote a paper with natural history and treatment of the muscle diseases. And it became a seminal paper in 1954, and people from all over the world came to Newcastle actually to be trained by John Walton. And so we launched our center actually 10 years ago with John being there. He died a few years ago, a few years later. So now I've mentioned neuromuscular diseases a few times, and I'll tell you a bit, well, what am I talking about? Okay, these are our patients, and I see patients with weakness. Okay, now traditionally in the history of medicine, people always thought if someone has weakness, a paralysis, it's the brain that is affected, like a stroke. Okay, in our patients, brain is working fine, but they have weakness, and these could be babies that can't move or can't breathe or can't swallow and need support accordingly. This could be adults, often people end up wheelchair bound or they need to be ventilated because the respiratory muscles don't work. And I'll show you just a few videos here. This young boy was asked to get up from the bench without using his arms. And so he's trying, but he needs to push himself up. Otherwise, there's not enough strength in his pelvic girdle muscles. And when he's asked to get up, he needs to turn around and push himself up, basically using himself as, and you can see this, it's called the Gowers maneuver after a famous British neurologist, William Gowers. This is a little boy here, and you can see walking upstairs is often extremely difficult, one step at the time. First sign of weakness in your lower limbs, or sometimes you see that patients have a waddling gait, looks very cool, but it is actually weakness. Or this gentleman, whom I had known for many years, has a form of limb girdle muscular dystrophy as well. And in order to get out of his car, he and he's an engineer. He actually developed this cushion that is self-inflating and pushes himself up. His wife bought a sports car, and he said to me, "But I want to drive this car, but I can't get out, so I need to come up with something." And so he actually was able then to drive a Porsche as well. Now the molecular structures, this is, or the anatomical structures, so this is the brain, and many of you will have probably heard about our motor cortex in the so-called first motor neuron. And this is all central nervous system. This has nothing to do with the patients that I see. The patients I see have a problem in the second motor neuron, which is blue here, or the muscle itself. This is a cross-section through the spine, and this is called the anterior horn, and the second motor neuron is called the anterior horn cell. So this structure is really responsible for maintaining muscle, for generating force, for reflexes. So the four structures, again, that are involved here is anterior horn cell, the axon from the motor neuron, where the nerve meets the muscle, the so-called neuromuscular junction, and the muscle fiber itself. Okay, these are the four anatomical structures. And if there are lesions in any of those structures or molecular or genetic defects, you have weakness. And when I see a patient with weakness, the first thing that I need to decide, is this acquired or is this genetic? And I'll just give you a few examples. So a genetic disease that affects the anterior horn cell, or the second motor neuron, is called spinal muscular atrophy. I'll talk more about this. Many of you have heard about polio, which is an acquired disease. It's a virus that can affect the second motor neuron. Or hereditary neuropathies. Some of you will have heard about diabetic neuropathy, which would be acquired. Or congenital myasthenic syndromes, and an acquired disease would be myasthenia gravis. Muscular dystrophies are all genetic. Inflammatory muscle diseases are acquired. Now, you may say, never heard of any of those diseases. And the reason why you may not have heard of them is because all of them are rare diseases. And in Newcastle, we are particularly proud of our rare disease research. And rare diseases means that less than one person in 2,000 people is affected by that disease. Sometimes the disease is so rare that there's only 1 in 100,000, or 1 in 1,000,000, or 1 in 10 million. This is where our expertise comes in, diagnosing those patients. And 75% of rare diseases affect children. 1 in 17 is affected. So there will be people here in the room that most likely have a rare disease. 30% of the kids that are affected with a rare disease will die before they're 5 years old. Many of them die as neonates on the intensive care unit. The complexity is that there are 7,000 different rare genetic diseases. Most of them are genetic. And it often takes 5 years to diagnose a patient from the onset of symptoms to actually getting a diagnosis. So I said there are 2 diseases I want to talk a bit about. You just need to keep them in mind. I don't go into too much detail. Spinal muscular atrophy affects the anterior horn cell. What is important is our neurons, once they are there, they don't divide. You will have heard that if you lose neurons, they don't come back. All the genetic information of a neuron is stored in the nucleus, and this nucleus doesn't divide. Now, the muscle cells in muscular dystrophy affect the muscle. They have many of these nuclei. We call them multinucleated cells. And the genetic information is in each of these nuclei. These are actually the 2 biggest cells that we have, motor neurons and skeletal muscle cells. Now, skeletal muscle is quite amazing. Any movement that we do actively in our body is driven by muscle or muscle proteins. And the biggest muscle is our buttock muscle. The strongest muscle is our masticatory muscle, the masseter muscle. Many of these are not just there to generate force, but to generate temperature. Our body temperature is mainly regulated through muscle as well. But it's also that the heart, from a myologist's point of view, is just a muscle. Cardiologists see this slightly different. Smooth muscle in our gut movement, all of this is driven by muscle cells. Actually, in each of your hairs, when you get goosebumps and the hair stand up, each single hair has a muscle that pulls up the hair. Okay? Any active movement is done by muscles. Now, because I'm a geneticist, I need to also, I've given you a bit of background about neuromuscular diseases. Now I give you a bit of background about genetics. This is not going to be complicated. So each cell has a so-called nucleus that contains our entire genetic information. And we call the entirety of the genetic information our genome. And the genetic information is stored on chromosomes. It's not really important, but you have heard about chromosomes. And the chromosomes are made out of DNA. And again, the chemistry is not really that important. But what you need to understand is a gene is like a construction plan, a blueprint for our cells, how to make substances, which we call proteins, that are important for the cell's function. Okay? So this is a gene here that's making an antibody. This is an antibody. So this is a protein. This is basically the central dogma of molecular biology. There's also a step with RNA in it. It's not that important, but that's the idea. The gene is a construction plan, like a recipe, and it makes a protein that is important for the cell's function. Now, DNA, I don't want to go into chemistry, but what is important, and you will probably have heard about this, DNA is encoded with 4 letters, basically. This is the chemistry, but it's 4 letters, really. You could say, it's not just an analogy, that's what it is. And it's A, C, G, and T. And what's difficult to comprehend is how big the entirety of our genetic information, our genome, is. Because our genome has about 3 billion letters. And we don't have a good feeling, what is 3 billion letters? Well, the Wellcome Trust has done this and has said, if we write the entire genome into books, chromosomes, from chromosome 1, 2, 3, 4, up to chromosome 22, and then there's the X and the Y chromosome. And we just use those 4 letters. This is how much 3 billion letters are. So that's 1 human genome. And if I take 2 people from the audience and would say, well, how similar is their genome? All of us here are basically identical. The only difference that we have is that 1 in 1,000 letters is different. C might be replaced by an A or by a G. If 1 in 1,000 is different and you have 3 billion, you can do the math, it still means that there are 3 million different letters between 2 people. So we are almost identical, but we are also quite different. This is why all humans on Earth look different except for monozygous twins. Now, not everything in the genome is yet fully identified, and this is part of our challenge as well. And just to illustrate this again, I mentioned spinal muscular atrophy. So this is where the motor neuron is affected. And in these babies, they don't have a protein that is called survival motor neuron protein because the SMN1 gene doesn't work. And if you don't have this survival motor neuron protein, your motor neuron dies. And these babies are floppy. They often have difficulties to breathe, to swallow, to move. And most of them die within the first year of life, within the first 2 years of life. It's the most common cause of a genetic disease for neonatal death. And this is the Duchenne muscular dystrophy gene. It's called Dystrophy for muscle fiber function generating force. I'll tell you a bit more about this. Because this is a disease that I've done my PhD in, and it's the best characterized, most known neuromuscular disease across the world. And it was first described by a French neurologist, Duchenne de Boulogne, in the middle of the 19th century. And he looked at muscle and he said, well, if this is normal muscle, and muscle fibers are long tube-like structures, and if you do a cross-section, this is how it looks. And this is when you take a muscle biopsy, a muscle sample from a boy with Duchenne muscular dystrophy. They often have a curvature of the spine, they have very prominent calves. The muscle will slowly waste, and most of them, if you wouldn't have any intervention, would die in their teenage years. Now, the average age due to better care is up into the mid-20s, early 30s. And they die because the heart muscle and the breathing muscles stop working. Duchenne gene is the biggest gene known in nature. And it takes about 16 hours for the cell to make dystrophin, the protein product of the gene. And the 10 books are here because it's so big. And again, if a normal page in a book has about 250 words, and if you take a book with 200 pages, then you have about 50,000 words, and a word on average has about 5 letters, so that's 250,000 letters. So the Duchenne gene is about 10 books. That's massive. And when the gene was identified, it wasn't clear what is the protein product, and it was very exciting to then show, oh, this is dystrophin. It is labeled here in red with an antibody and a red fluorescent dye, and it's surrounding the muscle fibers, protecting the muscle fibers. And in Duchenne muscular dystrophy, if you look for dystrophin, this is how it looks. It's gone. And this is why the muscles are permanently damaged, even if you have normal movement. Now, the question is, how do we actually identify a genetic change? Well, we basically take genetic information from a patient and then compare it to the genetic information of a normal person. But I just mentioned that we are all different, so ideally you don't want to compare to just a single normal person, but to a whole library of healthy people. And this is what we are doing with databases. If we know the gene that might be responsible for disease, we will compare it to data sets of healthy people. And then if we see changes, then we say this is possibly responsible. And it's not like finding a needle in a haystack. It's more like trying to find a straw in a haystack. That makes it difficult because finding the needle is actually easy now for computers and so on. But finding the straws is still challenging, and this is where our expertise comes in. Now, when I started, there were, in 1990, there were about 8 genes. One of them was the Duchenne muscular dystrophy gene. And with better technologies, this number, and this is for neuromuscular diseases, increased steadily. And this is just from March 2025. We now have 697 different genes that are responsible for about 1,240 diseases. Some genes can also cause different diseases. So, but even though I said I'm a geek and I'm a super expert, I haven't seen all of these diseases because some of them, there are only 10 patients in the world. But with new technology, the complexity, solving the complexity has become easier. We have now machines. Many of you may have heard about the Human Genome Project, which was at the time the biggest science project in the world. It was funded with about $3 to $5 billion US. It took 13 years to come up with the first single human genome, and there were centers around the world that were involved in this. And now I can do the same in our lab in 24 hours for less than £1,000. That's the technology that is unbelievable. And the public often doesn't really know what has happened in terms of technology, particularly when it comes to genetic information. So now this was part of my introduction, and I will now talk a bit more about, well, why Newcastle? Why are we good in neuromuscular diseases? And normally this is the acknowledgement slide, but I want to also emphasize that none of what I'm telling you would be possible without an amazing team of people, and it's very interdisciplinary. These are doctors and nurses and physiotherapists. These are basic researchers, but these are also people who know how to coordinate large projects, project managers computer scientists. So it's very interdisciplinary. And our center, which is based at the Center for Life, is working across the university and the Newcastle Hospital NHS Trust with experts in neurology, in respiratory medicine, orthopedics, imaging. So, Andy Blamire is an imaging expert with whom we work closely together. Mitochondrial diseases, there is a whole center for rare diseases in Newcastle. Cardiologists, endocrinologists looking at bone health, pathologists, and as I said, computer scientists, electrophysiologists. So it's very interdisciplinary. And Newcastle, since John Walton, has a critical mass of researchers and clinicians and healthcare professionals in neuromuscular diseases. I also want to show our funders because it reflects a bit the collaborative nature of our work. So there are pharma companies, Sanofi or Roche, very, very big pharma, biotech companies as well. There are many patient advocacy groups as well, the Muscular Dystrophy UK or the French Muscular Dystrophy Association or the Jane Foundation in the States. And then there's, of course, government funding, and I'm particularly proud to say, actually, that for the last 20 years, we have been generously funded by the European Commission as well. These are grants that we have been involved, have a volume of about €500 million. We only got about 5%, but that's still very good. So the teamwork and the funding and working with patient advocacy, all of this is really important. I will briefly start with the gene discovery because now you understand genetics. So I said, if we have a patient that comes with weakness and I decide it's genetic and not acquired, then I want to find the gene that is responsible for the symptoms. And as we said, we compare this with databases. Sometimes it happens that we check all the genes that are known to cause a neuromuscular disease, and this patient does not seem to have a genetic change in one of the known genes. Now that's particularly exciting for us because we think maybe this is a new gene or a new disease. The disease has always been there, but it has never been characterized or identified. And so this is a bit what our colleagues looking at the computers do is, spot the difference. Because I said all of us are different, and how do you find the genetic change that is responsible for the disease rather than just for the individual trait of the person? And so I'll give you a few examples just about our work. This is a project that is called MyoSeq, which was funded by industry and also by patient advocacy group, where we looked at 2,000 patients with muscular dystrophies across Europe. We asked those centers to send us DNA, and we would do the sequencing, and we would do the spot the difference and analysis, trying to identify genes. And in this project, we were able to provide more than 1,000 patients, and that means more than 1,000 families, because there might be more siblings affected and so on, more at-risk patients, with a precise diagnosis. So extremely rewarding. And what we do is we ask the centers to send us DNA. We process them in Newcastle. We sequence them with colleagues at Harvard University. We did the data analysis and then reported the findings. And because this was so successful and we identified many rare diseases and new diseases, the industry that is working with rare diseases also said, well, you diagnose patients for us and every patient counts. Could you do this across the world? And we now have a project that is called LatinSeq with Anna, who coordinates our sequencing, and Jordi, who is a colleague who leads this project, and Chamo, who actually produced the slide, where we do the same that we did in MyoSeq in Europe in Latin America. And here we now also have about 1,000 patients that have already been enrolled. 64 have been diagnosed. The ones that are not diagnosed are the ones that we like as researchers, because that's where we find new genes and try to understand pathomechanisms. Now, pathomechanism is, once you have a new gene, what's next? So as a patient with weakness, and we identify this patient has a genetic change in a gene that has never been described before, and we think that this is causing the problems, then we have to check in a genetic database, basically, well, has this gene been associated with other diseases before? Or has it never been assigned to a disease, which is good? Is the gene actually active in muscle? If it's not active in the muscle, if it's only active in liver, it will probably not cause weakness. How does the muscle pathology look from the patient? And if we then find that a protein is not active or expressed, that's a strong indication. And then we also, once we have a protein, we look at protein function. We do use animal models, particularly fish and mice, but also more and more cell cultures to understand the disease mechanism. Why is this causing weakness? Now, I just use this as an example. You don't need to understand this. So there were patients in our clinic, and we looked by imaging at their muscle, and their muscle, which is normally gray, was replaced by fat, and the biopsies looked very dystrophic. We identified 2 more families through international collaborations. All of these patients showed a very similar picture. We then identified a new gene, it's called POPDC3. We could prove that it was indeed affecting the protein, and then we tested all of this in zebrafish embryos, and these zebrafish embryos, we can induce the same disease, and we can also do functional analysis. This is just an example of research. You don't need to understand the mechanism here. But what is important is that we publish this, and we inform the world and say, we have identified in Newcastle a new disease for patients that present with weakness, and so that colleagues across the world can then say, well, I have patients that don't have a diagnosis. I will now check in this gene. This is how it happens. This is, for example, a family. Sometimes this takes decades. This is a family and a patient that John Walton saw in 1969. And he said that, quite frankly, the only way to clarify the muscle weakness is to do detailed investigations. And we provided this patient 54 years later with a diagnosis because we collected families across the world, 30 families. This is actually our patient here that John Walton saw. And he had a genetic change in a gene that we called S. But then there were other males that also had genetic changes in S that didn't have the disease, which is the black boxes. And it took us a few years to realize that this is our patient again. They also had to have a genetic change in a gene that we call T here. And the disease only manifests if you have mutations in 2 genes, which is very rare. And we, again, then published this around the world. And I have had feedback since last year that around the world, clinicians have now diagnosed patients with this new neuromuscular disease that we identified here in Newcastle. Now, all of our research is then something that we apply for our diagnostic service, as I just mentioned. So we do the clinical evaluation, we see patients, we do the genetics, we look at the muscle, we look at muscle imaging, might do more functional work, and we also have established in our team patient registries, because often patients say, so now I have a diagnosis, but how do you remember me when there's a treatment? How do you contact me? And that's when we say, well, you should register, and then we don't forget you. Because of all of our expertise here, NHS England has actually commissioned us nationally to diagnose patients with muscular dystrophies here in Europe. Even though you could say, well, east of England, our catchment area is about 3 million, but we see patients from all over the UK that come with weakness and that are undiagnosed. And we particularly also apply, and we are very, very good at this, world-leading muscle imaging. So, you see here a cross-section through a thigh and a cross-section through a calf. And these are all different patients. And even though you may have never seen a muscle MRI in your life, you can appreciate that somehow they all show different patterns. And we are now using pattern recognition software and AI to actually help us making a diagnosis. We are also trying to quantify the changes over time, so there's a big effort within our research. We are actually now involved in one of the EU projects where we use muscle, where we use genetic information, and where we use muscle imaging, and just ask computers with artificial intelligence to come up with a diagnosis. So genetic testing is now something that is also more and more considered to become more routine. And there's even a question, well, should we not start sequencing either new people at risk, so that when they have, they know they might get a child with a severe genetic disease, and they could do something about it. Go on a dating app and probably test genetic sequences. This doesn't exist yet. This is just my vision of what will happen. Now, but all of this, what I have told you about diagnostics and research, how does this make a difference ultimately to the life of patients? And this is where we then have to do clinical research. And we are currently involved in about 35 interventional studies. Most of them are commercially funded. And clinical trials are extremely challenging and extremely expensive. And if you start with about 5,000 to 10,000 potential drugs and you test them in a lab, animal models, you might end up with one that gets approved. And the timeline might take up to 15 years. And we are particularly strong here in this gap between the lab and then bringing this to the patient. And the different trial phases are often in healthy volunteers or a small group of diseased patients, and then getting marketing approval. And it says here that a trial could take 15 years and could cost about $1 billion. So it's a huge endeavor, and it's great to see that we are one of the centers that industry is working with. Because it's genetic diseases, often patients had asked me 20 years ago, so if I have a genetic change, can't you just replace the faulty gene with a healthy gene? And it's a brilliant concept, it's very easy to understand. So the normal gene makes a protein, you have a gene with a change, there's no protein, you introduce a new gene that makes protein again. And so the challenge was always, well, but how do we get the genes, the healthy genes, into the cells? Now, since COVID, everyone is a virologist, and everyone knows that viruses basically deliver genetic information, how to make a virus, to cells. Okay? Viruses aren't living organisms. They need a host to produce viruses. But what they do have is their genetic information. This is how you make a virus. So the COVID virus goes into cells and introduces DNA. This is how you make COVID viruses. And this is when researchers said, well, maybe we can use this, because we know, and all of you know, viruses also have a specific affinity to a tissue. Some viruses just go to the gut and you get diarrhea. Some of them go to your chest and you get pneumonia, or some go to the liver and you get hepatitis. So you can also target them. And the virus that we now use to deliver genes is the so-called adeno-associated virus. It's a fairly small virus, but the good thing is that many of you will actually have been infected without knowing, because it doesn't cause any symptoms. And once it enters the cell, it actually delivers the genetic information outside of the chromosomes. Now, the problem is that it is not very big. And the Duchenne gene, for example, would never fit into the AAV, the adeno-associated virus, which is a problem. So you can make the gene smaller. It's actually possible in terms of Duchenne. And so you need a smaller transporter, because that's the AAV. And you provide them with the GPS system and target them. Okay? This is the principle of gene delivery. And here in Newcastle, and this is my wonderful colleague Emma Heslop, who coordinates a network of trial sites for Duchenne muscular dystrophy. And so through this trial site that we have set up to support clinical research in Duchenne, Charlie was actually the first patient in the UK with Duchenne muscular dystrophy who participated in a gene replacement therapy trial. We are still waiting, because this is all fairly recent, to see whether this new therapy will be approved in the UK and in Europe. Now, it's not always successful, these clinical trials. Most of them actually fail. But I want to end with a success story. And this goes back from Duchenne to spinal muscular atrophy. I mentioned that these babies have problems with their motor neurons and therefore they can't move. In the type 1, the severe cases, this is the age, the survival rate. Most of them die within actually the first year of life. And by 6 or by 10, all of them have died. And so these are just 3 examples of babies with SMA type 1. They are often ventilated because they're too weak to breathe. They can't sit independently. They're often also tube-fed because they can't swallow. And over the past 10 years, we have worked with 3 pharma companies, Roche, Biogen, and Novartis, to think about increasing the protein that they are missing. And I mentioned at the beginning it was called the survival motor neuron protein. And we know that if you have more, then you have a milder form, which is called type 2. And if you have even more, you have an even milder form called type 3. So the idea was, can we increase the protein? And the best way to do this was, can we use a virus, introduce a gene, can we get it into their spinal cord? And if you do this early, actually the virus, that specific virus, goes into the spinal cord. And once it's in the motor neuron, it delivers its genetic material. And so this is Freddie's story. And so Freddie this is his first birthday, and he's sitting on his birthday cake here. Now, he can't really sit upright. He isn't strong enough for this. And this is when, a few months later, when he had his first wheelchair. And this is at 15 months of age. And you can see that he has no head control. If he drops his head behind his wheelchair, he can't get it up again. He's too weak for this. And so Freddie's mom works in our team. She's a nurse in our team. And she says, and she knew that we were doing clinical trials in spinal muscular atrophy. And she said, can Freddie be enrolled? She said, of course, we'll do this. And he was treated when he was 16 months old. And with his disease, he was at the stage when, before treatment, unable to have any head control, and he was unable to sit. And we provided him with the treatment, and this is a video of Freddie. You can still see that he is actually very weak. So his mom asked him, come, Freddie, sit up now. And he says, how annoying, and it's an effort. But what you can already appreciate, he does manage to control his head. And yes, it takes a little time, but he can push himself up and he can sit upright, and it's amazing. Now, then his mom, Steph, and she doesn't mind that I mention her name, became pregnant again. And we knew that when we tested the baby that it's spinal muscular atrophy again. And she said, but I want to keep the baby because we had learned if you treat really early, you can be more efficient. And so we prepared everything for Louis to get gene therapy at 2 weeks of age. And so, Louis would normally not be able to really sit independently or just, and this is Louis at about 14 months. He had gene therapy when he was 2 weeks old. This is Louis. And he's walking like a normal little boy, and he's lovely. And the video that I like particularly is this. This is when Louis is 2 years old. And now he's able to actually hold his entire weight as well. This was a boy with spinal muscular atrophy, which is, I work as a medic for 35 years. This is the most amazing thing. This is close to what we saw in the acute leukemias in the 80s. And these babies are now treated with gene therapy, and they basically, what these graphs show, is show normal development. If you treat them right after birth. We are now even thinking about treating them intrauterine. This is Louis at 3.5 years. Just 2 weeks ago, this is the family, and with Freddie, who was treated, too late to have the same amazing outcome as Louis, but it's fantastic. Now, 5 more minutes. So we have drugs now, okay? So it's from nihilism to treatments. The challenge is that some of these drugs that we are dealing with are the most expensive drugs in the world, okay? The treatment for Louis is provided by the NHS. It costs 1.79 million pounds. It's a single injection of viruses that deliver the SMN gene into his motor neuron. And as far as we know, he's developing normal. We do not know the long-term outcome, but once the genetic information is in the motor neuron, I mentioned it before, the motor neuron is not going to divide anymore. It's in there. But what we don't know is, well, is it good to be in there forever at high levels? Well, so we have to follow up these patients for, according to regulators, for 15 years at least. We will always see him. There's another drug for Duchenne muscular dystrophy, a gene therapy that's costing in the US currently 3.2 million, single injection. So that's a difficult discussion, the value of life. And our team is very closely discussing this with the payers, NHS England, which is advised by NICE. So they come to us as well and say, well, what kind of benefit do you see? What is the price tag for life? And there are different numbers. But in health economics, you need to talk about this. How many additional years of 100% quality of life can you achieve? This is the unit, basically, that we, come up with, or that is established when we come up with drug prices. So finally, I just want to say, what I've taught you today is, of course, also something that we need to disseminate to our colleagues in the world. So it's very important that we have a program of education and dissemination. And 2 of my colleagues, Michela Guglieri and Henrietta van Rooyen, have been involved in also doing this with the Royal Colleges here to inform clinicians how to detect neuromuscular diseases early, because from Louis we also know the earlier you treat, the better. And then final, and this is my last slide here, we are also making sure that everything we learn and where we have evidence for is then published in care guidelines. And this is just an example for Duchenne muscular dystrophy with 2 of my colleagues, Michela and Kathy, leading this project nationally, so that GPs also know these are rare diseases and what are we supposed to do, that they have care guidelines for this. So I hope you got an idea why Newcastle is seen to be top in the world when it comes to neuromuscular diseases. I also hope that you didn't fall asleep, and so thank you very much for your attention.